Fred Cross. I run a laboratory at Rockefeller University in New York City. What I'm using this instrumentation for in particular is a new project on the Green Algae Chlamydomonas reinhardtii. My fourth time or something in making a 6,000 density replica. So this is the source, and this was just made actually from a 96 pattern that just got replicated 16 times with this instrument just to make something at 1536. And so the reason why there's squares is because those are inviable or poorly viable colonies in the original single pattern. The plan is to, in order not to have the project require quite as long as the yeast projects, because the yeast projects were each decade-long projects, many labs. Between the availability of better instrumentation to accelerate just getting the mutants to begin with, combined with, at the other end, deep sequencing to identify the responsible mutated genes, we should be able to go a lot faster. That goes in there. This is just a plate just poured by hand on a bench top, so nothing special with the plate, which we talked about. And this is all set up to go for this purpose and should now work, I believe. So what I was going to do is go down and do a little the dry mix thing to get a good representation of what's on each colony on the pins. And you are making the 6,000 pins. So that'll be nice, because then we won't have to do this 4 times thing, which takes a little time. That's it. So this, I don't think you can probably see anything on here, but there are, in fact, 6,000 extremely similar looking little dots on here, some of which you can see a tiny bit of green stuff, not much. But that looks pretty nice. So we'll see. Grow them up and see what happens. What we just checked today was the capability to go to 6,000 density, 4 times 1536, and they just came off the instrument, and we don't know how they're going to grow up, but they look good so far. They look happy sitting on the plate. I don't know if you can notice, but the density has gone up, because before that one source guy was kind of right in the middle, and now there are 4 of them, and that's the pattern. And so, like the reproducibility question you're asking about, I mean, so far this looks pretty good. There's 4 spots from one source, and they do kind of look the same. And there's 4 from another one, and we can find probably some that they have a kind of a range of how dense they are in the source. But anyway, it looks pretty decent, I would say. Nowadays, it looks like just about every cell, because we can see the cells in the microscope right after they've been printed, and you might have gotten an image of that. And you can pretty much see that all those cells, give or take, are viable and go on and make little, grow up and contribute to make the growth of the patch. That seems good. That's certainly true out of liquid, and it seems to be reasonably true agar to agar. At this magnification, this is about 3 millimeters. So that's pretty small. And so and they all have look like they all have plenty of room to grow in. So we'll see. So you see this guy that's low here. There are going to be 4 replicates of it, and I think they all are. So the reproducibility is really pretty good. There's another sick one. Column of sick guys. So far, the accuracy seems to be good enough that if it's instructed to hit a particular spot in that array, it will hit that spot and not some other spot, and better than a toothpick. So if that's true, and if that works out, that'll be a big help, because the factor of 4 that's a big factor.